Thank you so much, Navtev. Let me try and get to talk. So we're going to switch gears a little bit into the preclinical realm, and I'm going to give a research talk with some preclinical mouse models of an orthopedic implant infection, where I think we can learn about the pathogenesis, some insight into therapeutics, as well as new diagnostics. And if you stick with me, thank you. Well, if you stick with me and you think of these mice as patients, I think you'll be able to follow. And the room is small enough, so please raise your hand if you don't understand something. I'll be happy to answer as the talk goes on. So I'm actually a dermatologist, and I got into this field because the microbiome on the skin can infect any implant or surgical implant or anything from a catheter to a knee replacement or a prosthetic joint. The flora from the skin can go in and infect these implants and form biofilms, and they're a real problem. So in the U.S., there's about 1 to 4 million knee prosthetic joint replacements with knee and hip. That 1 million number is going to go up to 4 million by 2030 because of mostly because of the aging population and the more need for knee and hip replacements. Prosthetic joint infections are, thank goodness, not a very common problem, but they're around 1% of primary joint replacement surgeries. But on revisions, because of infection, that rate can increase to 3 to 5%. And they're a big problem because the treatment requires real extensive surgical and medical care. If you think about it, what you got to do is you got to basically take the implant out so the patient can't walk, and then you put the patient on 2 to 3 months of intravenous antibiotics. And during that time, there's a lot of morbidity or mortality due to immobility and disability, and the cost, because they have to take, in at least the United States, you take the implant out and you have to do another procedure to put the new implant in. It costs about $100,000 to treat one of these patients in the U.S., and that corresponds to a national health care burden between 1 and 3 billion annually just to treat orthopedic implant infections. The most common etiologic pathogen is staphylococcal species, including now these antibiotic-resistant strains we're seeing, especially in the U.S., including MRSA. And my work involves a small animal model to non-invasively use optical imaging. We basically use light to study these infections. So that's what I'm going to talk about today. I just wanted to show you some pictures of what a total knee hip replacement and a total knee replacement look like. These are very complex implants with multiple metallic and plastic parts. If you think about a bacteria getting in the mix of these, they can bind to any of these foreign surfaces and form a biofilm and really wreak havoc on the bone and cause a chronic inflammatory state, which causes these implants to fail. Like I mentioned, these biofilms form. This is a picture of a staph epidermidis biofilm. You can see the clusters of bacteria of staphylococci growing in clusters, and they're surrounded by a matrix. And these are very hard to clear. If you think about it, there's no direct blood supply to an implant, so the antibiotics can't get to the implant, and they can't penetrate the biofilm. So antibiotics are very ineffective. That's why frequently these implants have to be taken out. Here's an example of what you might see clinically on an x-ray. So this is an infected hip replacement. And around where the bone cement is, around this part in the femur, you see this, I don't know if you can see it in the back, but you can see these areas of osteolysis or bone loss, this radiolucent area. That is called periprosthetic osteolysis, which is really characteristic of these infections, because the patients don't come in sick. They don't come in with fever. They frequently don't have other signs of infections, but they will come in complaining of a loose implant. And the reason for the loosening is this periprosthetic osteolysis, so the implant is not as stable. So we tried to model this in a mouse model. A mouse joint is much, much smaller than a human joint, so we had to really use a very simplistic model. So what we basically do is we insert an orthopedic Kirschner wire. For those who don't know, it's a very small wire that's used to set wrist joints. It's about 0.6 millimeters in diameter. We cut them to about 1 centimeter in length. It happens to fit very nicely in a mouse femur, and we leave the end sticking out into the knee joint. And so we place this surgically, and at the time of surgery, we put a small inoculum of Staph aureus into the joint to cause a knee infection with metal in the joint. So it's a very simple model where metal is in the joint and in the bone, and you get basically a Staph aureus inoculation just as it might occur during the time of surgery or in the postoperative period. We use light. So this is an example of a bioluminescent strain of Staph aureus. So you can see the bacterial colonies on a plate. when you put it in an optical imaging machine to detect photons. Every colony on this plate emits light. And as long as the bacteria are alive, they'll naturally emit light similar to a firefly, except you don't have to have any substrate. It runs on bacterial aldehydes produced during metabolism. So as long as the bacteria are alive, they will emit this light signal. And we basically infect mice with this signal. And we use a lot of different transgenic mice. The ones I'm going to talk about today are these lyse EGFP mice. So this is an EGFP, a green fluorescent protein that is expressed on the lysozyme promoter. Essentially, the mouse has green neutrophils, green fluorescent neutrophils. And because with bioluminescence is light shining out and fluorescence, you shine light in and it emits a longer wavelength, you can actually discriminate these two signals so you can get an idea of the bacterial burden as well as the neutrophil recruitment. So we image these mice in an in vivo imaging machine. We happen to use this one made by PerkinElmer, which is basically a black box with a charge-coupled device camera to detect photons. And we use this to detect in vivo bioluminescence, so we can detect the bacteria coming out of that knee, and we can also detect the neutrophil infiltration at the same time. So it gives us a good idea of the bacterial burden as well as the host immune response. We also have other endpoints that are important clinically. We look at biofilm formation on the implant we put into those mice. We collect bacterial CFUs, so the bacterial colony forming units that are on the implant and in the bone and joint tissue. And then we also measure neutrophil influx as a second measure, not just using the light. We actually take the samples out, homogenize them, and look at histology and myeloperoxidase assays, which is an enzymatic read of how many neutrophils there are in the tissue. And we also tried to really look at this periprosthetic osteolysis to see if we develop that in this mouse model. So here's an example of what the data look like. So if you put just 500 bacteria in, we can actually see the light signal in the knee joint. This is a short experiment. We ended in 5 days. If you don't infect the mice and just put the implant in, we don't see anything. This is a very sensitive technique, light, because nothing else in the body gives off a light signal. So the only thing that does give off light is the bacteria. So you can actually quantify that compared to background, and you can get a detectable signal. This is what the EGFP neutrophil signal looks like. So you can see at the time of surgery, right after surgery, there were pretty much no signals. And you could see in the infected mice, there's much more fluorescent signal compared to the uninfected mice, demonstrating the infection is causing more neutrophil infiltration. Any questions about this? Okay. All right, so one of the good things about this technique is you can monitor difference between individual animals. And this infection actually never clears. So this is 7 weeks of an infection with this certain strain of bacteria, bioluminescent Staph aureus. And basically, it stays above background for all 7 weeks of infection. And these two mice did the same thing. So you can see signal pretty much through all the 7 weeks. And you can detect that in the blue line here over that course of infection. This is what the fluorescence signal looks like. So the fluorescence neutrophils seem to taper off within 2 to 3 weeks. And that's what happened here in these two groups of mice. So at the time of surgery, it caused a lot of inflammation. These were all infected, but that signal does dissipate, even though these mice are chronically infected. And here's a mouse that did something different. So in this case, the signal in the knee joint increased over time. It didn't kind of decrease. And here we saw actually very little neutrophil influx. So it gives us this in vivo imaging gives us a chance to look at differences between certain animals that may be different from one group to another. So this is what some of the histology looks like. So you can see in an infected joint, this is 1 day post-operative. You see inflammation here right in the knee joint tissue. If you don't infect the mice, you don't see anything. I'll show you what that looks like on higher power. We saw all these blue cells, which are neutrophils. We also see gram-positive bacteria in the joint tissue. Under higher power, this is seen much better with the blue cells of neutrophils and the blue bacteria seen on gram stain, the gram-positive Staph aureus we put in the joint. And if you don't infect the mice, basically you don't see any, very, very little inflammation, and then we didn't put any bacteria in, so it's sterile. And then we want to see if there were biofilm formation. So before we put the pins in, this is what the end of the cut pin looks like. It basically looks like cut metal on scanning electron microscopy. That's at higher power. And if you don't infect the mice and take that pin out at day 7 and 14, it basically looks the same. It just looks like cut metal. If we infect the mice, you get basically an ice cream cone of biofilm and material formed on the end of this implant. It's much more well formed, and you could even see those clusters of bacteria. So you really get the whole surface of the tip of that pin in the knee joint covered with the biofilm. We spent a lot of time searching the globe for a bioluminescent Staph aureus strain that would work well for us. We found one of these strains called made by this company Xenogen What was amazing with this strain, no matter if we injected with just 100 bacteria, 1,000, or 10,000 bacteria, which is all the curves here, they basically went to a steady state level by about 14 days and all produced the same amount of light. It was very bright, about a log higher than background. And you could see the difference between 100 bacteria, 1,000 bacteria, and 10,000 bacteria. They all kind of had the same amount of light, and that was a really strong signal. So this seemed to be a signal that we could really follow the mice chronically over time. So one of the things we did with this model is try to determine optimal antibiotic therapy, which you really can't do well in patients because only 1 to 2% get infected, and you have to do thousands of patients to really see statistical differences. One of the things we were interested in is comparing vancomycin, which is kind of the standard of care in some of these MRSA infections, and I'll show you how we did this. And we compared it to vancomycin plus the second antibiotic, rifampin, which is very good biofilm penetration and is now recommended clinically in patients. So we wanted to confirm if that was indeed the case in mice. So what we did here is we infected 3 groups of mice, either a sham, of vancomycin or vancomycin rifampin, and we let the infection go for 1 week. And then we started these antibiotics. And you could see the sham, which basically got no treatment, had a bioluminescence signal more than a log higher than background through 6 weeks of therapy. The vancomycin alone group knocked this down a little bit, but it didn't really it didn't go to 0, so there was still plenty of bioluminescent bacteria there. You can see that here. And then when we added rifampin, this went down to background levels. So the addition of rifampin made a huge difference in the bioluminescent signals. Did this actually treat the CFUs? So what we did is that day, 6 weeks after therapy, we got the number of CFUs in the bone and joint tissue, homogenized it, and count them. There was over 100,000 in the bone and joint tissue. There was over almost 1,000 on the pin itself. And in the vancomycin-treated group, there was a very slight decrease in the bone and joint tissue, more decrease off the implant. But when we added rifampin, we almost sterilized the tissue and completely sterilized the pins. So the addition of vancomycin and rifampin really made a big, big difference. We wanted to look at the x-rays of the bones to see what they looked like. So you can see how much bone change, and we measured this in a width, occurred in an uninfected mouse. And then actually the addition of vancomycin alone, even though it didn't really decrease bacterial burden, it preserved the bone. And when we added rifampin, we also got preservation of the bone. And both of these look very similar to a completely uninfected mouse. So the antibiotics really did a really good impact on preserving bone architecture. Now I want to switch gears and see if we can really look at that periprosthetic osteolysis, and there's a way to do this in this mouse model that simulated what they're seeing clinically around those implants. So we basically looked at this other imaging platform, which was Illumina XR, which was an X-ray machine on top of our box. So here you can take an X-ray of the mice and see where the signal is. So here now you can see the femur and the tibia. We put the infection in here, and you could see the infection over time in respect to the bones. This is the EGFP neutrophil signal you can see. And this was an infected animal. An uninfected animal, there was no bioluminescence, and there was some inflammation due to the surgery that dissipated faster than the ones that were infected. And we really tried to see if we could measure periprosthetic osteolysis. And in the infected mice, and this is at day 48, you could see some abnormalities of the bone, but the resolution really wasn't enough for us to make any real good conclusions about if we are developing periprosthetic osteolysis. So what we did is we had to combine 2 other imaging platforms with a Spectrum, which is a big optical imaging machine with very high resolution and can do 3D imaging of the light. And then we used this quantum FX micro-CT machine, and we could take the chamber out of one machine and put it in the other machine and try to co-localize the signals. And so that led to us to make some pretty cool movies. I'll kind of show you an example of one, where now we could see in 3 dimensions that implant in the femur. And at the tip of the femur is that bioluminescent signal in 3 dimensions, right where we thought the bacteria infection was. So right at the distal femur and in the knee joint, we're seeing the infection. With 3D micro-CT imaging allowed to do these 3D reconstructions, so you can see the uninfected mice had very little change in that bone over 48 days, where you can see marked changes in the bone in the infected mice. And what was really interesting is the bone volume in the infected mice actually doubled over that course, where the bone volume only slightly increased as the mice grew over the course of those 7 weeks. I'll show you what the bone, more cuts of the bone. So here's what the bone looked like in uninfected mice. Kind of the implant or the bone really hugged the implant when it was uninfected. And you can see it in cross-section. Here's the implant. Here's really good cortical bone, high-density signal surrounding the bone, showing really good osseointegration. When you infect the bone at 14 days and at 48 days, you started to see really dramatic changes in the bone, and really the bone was increasing in size and splaying away from that infected implant. And if you look to see what that looks like in cross-section, is at 14 days we were already seeing radiolucent areas surrounding the implant, suggesting periprosthetic lysis. And this bone here, we saw more areas of radiolucency, suggesting what they see clinically. We can see in this model. And this actual volume of the bone is double the volume of this bone. So the bone started to expand in size with the infection and inflammation over time. This is what that looks like histologically. So this is an uninfected implant. You see a really good femur. This is where the implant would be. We had to pull the implant out to make these images. This is the tibia. This is the knee joint. Really very little inflammation. At 48 days, you can see how deformed this bone looked, and this corresponds to the increased volume we were seeing radiographically. And you can see here in the cortical bone, we were able to detect some blue bacteria in abscesses. You can see it much better here inside the cortical bone. These have been described in osteomyelitis as Brody's abscesses. We were actually able to see these in the cortical bone in all of our mice that got infected. So we really developed a septic arthritis, a biofilm, and an osteomyelitis in this model. We didn't see anything in the uninfected mice. Okay, now I want to end this talk with, I'm really using light here, but can this be used more than preclinically? Can this be used in patients? And one way possibly is through diagnostics. And this is using really the latest technology in fluorescent probes and a new technology called photoacoustic imaging. So this is basically what we wanted to do here is we tagged an antibiotic, and we tagged 2 antibiotics that are proprietary, so I can't tell you which ones we tagged, to ICG, indocyanine green, which is used in fluoroscopy in humans, is FDA approved. This is a near-infrared dye, and it can penetrate deep into tissues because the longer the wavelength, the longer it goes through tissue. I don't know if I could demonstrate this, but like if I show you like green light is a very short wavelength. You can't see it go through tissue. But if you use a red light can go right through. So the longer the wavelength, the more chance it has to go through the tissue. Sorry. So our control mice is we just injected ICG, the ICG alone. So this is the bioluminescent bacteria in our model. We injected ICG. We measured the fluorescence to see if it went near the infection in the implant. We didn't see anything. But when we did an antibiotic that's targeted against the gram-positive bacteria and tagged it to ICG, it went to right to where the infection was. So we saw the bacteria, and now we see our fluorescent probe actually go to where the bacteria are. So that can suggest that there's an actual infection by using an antibiotic targeted against gram-positive bacteria with this fluorescent probe. We repeated that with a different antibiotic, also tagged with this ICG probe, and we could see the fluorescence basically accumulate where the infection was. So it gives us a non-invasive way to say, is this a gram-positive infection, and where is the infection? And this time it's active site of in the joint. Then we wanted to ask is, can we see this through another imaging modality? Because really light in people really can't go through more than a few millimeters, or 300 micrometers of skin about. But photoacoustic imaging can measure 7 centimeters deep into skin. Through the skin. So we think this might work in humans. So the way this technology works is it uses a laser to pulse the illumination into the skin. And then if you have an optical probe like that ICG, it'll take that fluorescent probe and give this thermoelastic expansion, which leads to pressure waves. And you can actually detect that with an ultrasound. No radiation needed. So it gives you a way to detect the fluorescence signal 7 centimeters deep into tissue. We demoed this new technology called this vivo laser system from Visual Sonics. So this gives us that laser as well as 3D ultrasound. And basically our antibiotic ICG is a very strong peak because it's a fluorescent signal over blood, over anything else. So we can actually see that peak very well. I'll show you what some of these images look like. Basically we're able to see this very light signal. This is our ICG fluorescence. With the femur, the tibia, and the fluorescence was all over the place. This is the inguinal lymph node. And I'll show you what this looks like with our probes. So the femur, the tibia, the inguinal lymph node, and popliteal lymph node, we actually got signal all over the place. So it wasn't as specific as we would have liked. But we did see a photoacoustic signal. This is the metal pin. And we did see signal right where we thought the infection would be, but we did see signal in other places as well. So it wasn't totally specific. So I'll show you what this looks like on 3D too. So this is first antibiotic. So this is ICG alone. Antibiotic 1 did not work well. The other antibiotic, we could actually detect the signal with the photoacoustic signal. So we actually saw signal, which was very promising to us. And it was around the site of infection. And we don't know, we think there's a lot of nonspecific signal going to the site of inflammation that's either picking up where there's more perfusion from the inflammation, and the dye ended up going there, which is why we saw a lot of nonspecific signal. And you could do this in 3 dimensions, so you could start to also make movies in 3D, reconstruct where you're seeing signal. So I think, this technology is very new, but I do think it does have the possibility to translate optical imaging and fluorescent imaging into a patient to help with diagnosing the infection and providing some insight non-invasively. I'd like to thank the members of my lab that helped out with these experiments, especially Yu Wang, and they were performed in collaboration with Nick Bernthal at UCLA. We have multiple other collaborators around the world, and thank you for your attention.